All right, class, welcome to Chapter 8 of Molecular Biology. Today we're going to talk about genes, genomics, and chromosomes. Okay, so let's talk a little bit about the structure of DNA, or the higher structure of DNA beyond the double helix. And so we're going to start at the bottom here. And so we know DNA forms this double helix of anti-parallel strands. And within this DNA, this is the types of DNA sequences here are listed. So we have single copy genes and gene families. We have exons and introns and intergenic DNA, so the DNA in between genes, mobile DNA elements like transposons and things like that. We'll get into some of this stuff later. Single sequence DNA or simple sequence DNA and sequence repeat DNA, such as microsatellites and mini satellites, etc. So we have all these elements that are in these double helices, the double-stranded DNA. Now, that is packaged into nucleosomes. So nucleosomes are proteins that wrap up DNA around them. And here they kind of use the analogy, it's like beads on a string. So they wrap up the DNA and they prevent access to it. So they kind of package it and prevent transcription factors and modifiers of DNA from being able to access that DNA. Then from there, those beads on a string, the nucleosomes, form chromatin, which are these even more highly packaged groups of nucleosomes. So the nucleosomes get wound together into these bigger 30 nanometer fibers. In addition, we have loops of those fibers. So we're getting bigger and bigger as we go here. And then we have higher order chromatin folding. So we get even larger loops until we get to those interphase chromosomes. So when you look at a karyotype and you see the chromosomes as those packed, long structures that have the centermere in them, that's the higher order DNA, the interphase chromosomes. But then when transcription and things like that happen, that's when DNA is unwound selectively to allow access to genes that need to be either transcribed, or when genes need or genomic replication occurs, the DNA needs unwound as well to allow access. So throughout the course of a species' natural history, there can be duplication of exons or duplication of genes. So if you're to look at a gene like globins, there are multiple different types of globin. You're familiar with hemoglobin, and there's multiple genes encoding for these different types. Another example would be kinases. There's a lot of different kinases, and a lot of these arise from gene duplications and then specification. So we'll duplicate our gene, and then each of those copies can go and evolve to be have different function or slightly different function. And so that leads to increased complexity of organisms. Here we're going to talk a little bit about exon duplication. And so the way that both of these arise is through unequal crossing over. And so in this case here, where we'd have two chromosomes, each of these genes has 3 exons that are labeled exon 1, 2, and 3 here. And so if crossing over were to occur naturally between L1 and L1, and this, I guess, is L2 and L2, then we would have no increase in exons for that gene. We just have the normal amount of exons. However, in this case, we have crossing over right here at L3, and that's not between L3 and L2, so that's not an equal crossing over. And what occurs then is we have one chromosome that we have exon 1, exon 2, exon 3, and then another copy of exon 3, and then the other chromosome only has exon 1 and exon 2. So we've increased the number of exons, or protein coding elements of DNA, in this gene. And another example of how we can get entire gene duplication is through crossing over again, but in this case we have unequal crossing over during meiosis. And so what happens here is if we have a crossover event that is supposed to be between L1 and L2, but instead happens between L2 and L1, which we have kind of in this section right here, what we do is we have a chromosome that's completely duplicated both genes, and then a second chromosome that is missing that gene. So if this is an essential gene, it's likely that the second chromosome here is going to lead to, if that were the sperm or the egg that was fertilized, it would lead to probably a non-viable embryo. But in the case of the top individual here that has a duplicated gene, unless there's a gene dosage problem, it likely won't have an effect on the embryo. And then having two copies of that gene can lead to one gene evolving a specific different function way down the line in its ancestors. Okay, so now we're going to look at genes in terms of alternative splicing. So we have this first gene here, gene A, which is a simple transcription unit. And this makes up roughly 10% of genes in the human genome. And so we have our control regions here, which may be an operator, a enhancer, promoter, etc. Things where transcription factors need to bind. And then beyond that, we have three exons, exon 1, exon 2, and exon 3. And all three of those exons are incorporated into an mRNA that we can see down here. And so if we were to have mutations in A or B, that would affect this gene in totality. And so whether it stops expression of the gene or leads to leaky or reduced expression of the gene, it depends where the mutation is and how the promoter and the operator and the enhancers are all set up. But we also have mutations that can happen in C here. And if that occurs, then that can lead to, again, it could be a silent mutation, a mutation that either changes a base pair at a wobble base and doesn't change the amino acid incorporated, or we could incorporate a similar amino acid so it causes no change in the protein's function, or it could break the protein. Then we have mutations in D, which is the intron here, and unless that intron is, or that mutation is at a splice site, a recognition site for alternative splicing or intron splicing of this gene, then likely there's not going to be any effect on expression of this gene. So that is simple transcription units. So like I said, that's only 10%. So a larger share of our transcription is this complex transcription. And this is where alternative splicing comes in. And so here we have our two regulatory regions again. And we also have 4 exons. We have exon 1, 2, 3, and 4. And so in our first mRNA here, we have all 4 of our exons being incorporated into that mRNA and then into that protein product. So if we have mutations in A or B, again, just like our simple transcription unit, that can lead to reduced expression or no expression. If we were to have a mutation in C here, that could cause, again, a non-functional protein or a reduced functional protein. But more importantly, what we want to focus on is D. So if we have a mutation in D, mRNA1 is going to be affected, whether it's a silent mutation or a protein breaking mutation that's going to cause it not to work anymore. That's kind of outside of the point right here. But it's going to affect that protein. Now, if we were to look at alternative splicing, mRNA2. mRNA2 has 3 exons incorporated. It has exon 1, exon 2, and exon 4. And so a mutation to D is not going to affect the second alternatively spliced mRNA because that exon is not even in the mRNA. And so a mutation can affect some of the alternative spliced products of a gene and not others. Let's move on to the next example here. So here we have an example of multiple end exons. And so again, we have the A and B regulatory regions. We have 3 exons in this example, 1, exon 2, and exon 3. And each of these, unlike the gene before it, have their own poly A tail site. And so in the second example, we have exon 4 is the only exon that has a poly A tail site to end the mRNA. But in this example, we have exon 2 and exon 3 that both have their own poly A tail sites. And so these 2 exons can't be added together, or otherwise we'd have an exon, a poly A tail, another exon, and a poly A tail. And so that's not going to work. But in this case here, we can then use exon 1 with our exon 2 poly A tail, and then exon 1 with our exon 3 poly A tail. So if we were to have a mutation in exon 2, we would affect only the first mRNA, and if we were to have a mutation in exon 3, we'd affect only the second mRNA. And so along that same line of thinking, in our last example here, we can have genes that have multiple cap sites. And so in this example, we have these control regions. Each cap site has its own control region, right? And so exon 1A, the first option for the starting exon, has its regulatory region, its own promoter, F. And cap site 2, or exon 1B, the second possible cap site, has its own regulatory region, G. And so if we were to have a mutation again in exon 1A that only affects the first mRNA, if we were to have a mutation in exon 1B that affects only the second mRNA. So a mutation in a gene, in the coding region of a gene, or even the regulatory region of a gene, is only, or can only be affecting one of the multiple differently spliced products of a gene. So here for your reference is the major classes of eukaryotic DNA and how much of the genome they make up. So protein coding genes only make up about 40% of the human genome, and there's the number changes I've seen between 21,000 to 23,000, some higher to 24,000 genes. And so it depends on the model you use, because these are all predicted with computer models. There's also long non-coding RNA genes, which are regulatory elements RNA that is not actually turned into a protein, but the RNA actually has function. We have tandem repeated genes. Ribosomal RNAs is one of these genes, as well as small non-coding RNAs. We have repetitious DNA, so simple sequence repeats that can be variable, about 6%, so microsatellites, things like that, as well as DNA transposons, retrotransposons. These are things that are mobile jumping genes. They used to be called mobile DNA elements, etc. And then lastly, kind of going back to our gene duplication, processed pseudogenes. So sometimes when genes get duplicated, instead of evolving to have their own function, sometimes they can just break apart and gain all these mutations and then not just kind of be artifacts of former genes or duplicated genes. And so those are still present in the genome. And then we have these intergenic regions, so regions between genes. Okay, let's look at gene density in higher and lower eukaryotes. So first thing I want to point out is that in higher eukaryotes, there is a lot more intronic DNA region than there is in lower eukaryotes. And so as you can see here in our common yeast, we have very little space in between genes. While you compare that to the human beta globin genes at the top here, we have big swaths of DNA that are non-coding for exons. They're all intronic or intergenic regions. As you can see, both of these are 80 kilobases long. And so the other thing I want to point out is the products of our gene duplication. And so when we talked about gene duplication, we talked about how these genes can be duplicated and then arise through selective pressures like drift, slightly different functions. And so that leads to these different isoforms. In the case of the human beta globin gene, we have 5 different versions. We have HBB, HBD, HBG1, G2, and HBE1. We also had 2 pseudogenes here that were at some point duplicated copies of a beta globin gene, but then has lost their function since then. And so these genes will have slightly different functions evolved over the course of natural history. And so in the case of humans, we have HBE1, and this is expressed during the embryonic stage of development. So when you're very, very small multicellular ball of cells, HBE1 is expressed. But as you continue to develop and you become a fetus, HBG1 and HBG2 are expressed, which give a little more specificity that and are more tuned to what the fetus needs from development. And from there, when you get closer to birth and after birth, you express the adult version, HBB and HBD, adult, fetus, and embryo there to mark for you. You express this adult version because it is more tuned to carry oxygen in an adult as opposed to the fetal or the embryonic stage. So in addition to genes encoding mRNA, and then those mRNA going and becoming proteins, there's also RNAs that are encoded that don't become proteins, but actually have function within the cell. And so we've talked about a few of these before. rRNAs are the ribosomal RNAs that help the ribosome to actually do the process of creating proteins. We have tRNAs that have the anticodon loop that match up to the mRNA and bring the appropriate amino acid. We have small non-coding RNAs, which are involved in mRNA splicing, in addition to U7 SnRNAs, which are responsible for the processing of the 3' end of mRNA. I'm not going to go through this entire list because there's a lot of them, but as you can see here, these are a lot of these RNAs that are not coding still have function. Just because they're not making a protein doesn't mean that every RNA that doesn't make a protein doesn't do anything. So there's roles for RNA outside of making proteins. Okay, so let's talk a little bit about these simple sequence repeats. So there's portions of DNA that have these motifs or sequences that are repeated over and over and over and over again. They can go by a lot of different names. Simple sequence repeats, microsatellites, mini satellites, they all have different definitions. A microsatellite is about 1 to 13 base pairs. Mini satellite can go up to about 500 base pairs, I believe. But they're all essentially the same thing, where they're stretches of nucleotides that are repeated over and over and over again. And these sequences are often very variable between different individuals, or at least between relatives. And so a lot of these can be used for genotyping purposes. And for instance, back in the day, when they would try to identify people for the purpose of forensic DNA, right? So they're looking at, is this person the criminal? What they could do is they could sequence these microsatellite loci and count the number of repeats they had at these different loci. And if you did this for about 20 loci, you could get to about the specificity of 1 in so many million people that would have that same fingerprint of microsatellites. And so that's how it used to work. Now I believe they use SNPs and technologies kind of moved beyond using microsatellites. But let's look at how these are generated quick. So in normal replication at the top here, we have this CAG sequence repeated over and over, and its complement GTC that's repeated n number of times, however many times. So what can happen though is we can get what's called backward slippage, and what occurs is these CAGs will line up with their pairs, with their GTCs, but we'll get this loop that forms. And because these repeats in front of it, these CAGs, are still fulfilling their matchmaking purposes, their bonds, this loop doesn't have a partner, and it doesn't, the DNA doesn't feel like it's missing something on the template strand here, right? Because it's already matched up with its CAGs, and they're holding this loop that's not in place in that position. So what happens is the strand continues to replicate, and we get a full mixture or a full pairing of the synthesized strand and the template strand with this bubble in place. And then the next time that replication occurs, both of these two strands, the 5' and the 3' strand that we looked at in part B, those both become template strands, right, for the next round of replication. So then when that occurs and these get pulled apart, the pink strand on the top has one extra CAG in it because it kind of pulls that bubble straight. And now this microsatellite sequence on the one daughter strand has an extra copy number or an extra microsatellite, so it's n+1 compared to the strand it was partnered with. So because of this, there can be variable numbers in different people because the slippage happens a lot, since those repeats are still able to fulfill their bonds when a bubble like that forms. So like I said, this can be used for genotyping purposes. They're very variable between like families, but not so much between like brothers and sisters. So they're preserved enough to tell the difference between, you know, you and I, but you might have the same repeats as your brothers and sisters and your parents. And so it's kind of in between variability. But also they can cause disease, microsatellites. And so there's 14 different neuromuscular diseases that are associated with microsatellites. Huntington's disease is one of them, where these microsatellite triplet repeats are encoding in an actual gene, and that causes this single amino acid to be copied or placed over and over in the protein, and it leads to aggregation of the protein over time that interferes with neuronal cell functions. So they can be damaging. A lot of the ones that are used for genotyping purposes are in non-coding regions of your DNA, but they also can be in genes. So I talked about how these molecular markers such as micro or mini satellites can be used in forensic DNA analysis, but it can also be used in paternity tests. So in this case here on the left, we have the mother, and then we have the child, and these are the microsatellites that they have. Remember, they are different lengths, and so if you were to put them on a gel, they would create different bands on that gel depending on how many repeats you have. So the child will have some of these repeats from the mother and some of them from the father. And so when you look at these columns, you can see that the child has this band here in common with father 1, this band in common with the mother, this band in common with father 1, and this band with father 1, etc. So if you were to compare that to father 2, which I'll do in a different color here, blue, you see that father 2 doesn't share those bands where these arrows are, where the child has it. So this person would not be the father of this child. And so now looking at the DNA in the forensics case here, so we have our victim's DNA, and then we have the specimen that was collected from the victim, whether that is blood or semen or whatever the case may be that has DNA in it. And we have 3 suspects, and so we run this microsatellite panel on this suspects, and you can see depending on the length, they will, of each microsatellite, how many of those repeats that individual has, they'll migrate different lengths on the gel. And so in the case here, we see that the specimen matches up completely for all those microsatellites we tested with suspect 1, while suspects 2 and 3, even though they might have a couple that are in common, like right here, or at least those two, suspect 1 and 2, they'd have to have all of them in common. And so we know that the specimen belongs to suspect 1. So let's talk a little bit about mobile DNA elements. These used to be called jumping genes. They're now called transposons or transposable elements. The discovery was credited to Barbara McClintock, who found these in corn, and now that we find them in everything, including mammals. So 25 to 50% of mammalian DNA is made up of mobile elements, and about 45% of human DNA. And so there are numerous copies of these relatively few sequence families that are interspersed throughout the mammalian genome. And they're kind of like molecular symbionts, and they have this unique ability to be able to move throughout the genome by kind of cutting themselves out and then inserting themselves in other places. And so there are two types of these mobile elements. We have DNA transposons, and then we have retrotransposons. And so what a DNA transposon will do is it will excise itself from the DNA. And so you see here the gray was the flanking DNA, and now it's missing because it's cut itself out. And then that DNA will find another site, and it will insert itself into that site. And so it's a jumping gene, as people used to colloquially refer to it as. So it's jumped from one part of the genome to another. And so a retrotransposon, though, has an RNA intermediate. And so what happens here is that for a retrotransposon, we have RNA polymerase, which will actually transcribe an RNA copy of this jumping gene. So we have that right here. And so instead of this gene here cutting itself out, it transcribes an RNA copy, and then reverse transcriptase comes in and converts that RNA back into double-stranded DNA. So this is that same enzyme that we use when we're creating cDNA libraries to turn RNA into double-stranded DNA. So it naturally occurs in the cell as well. And so that will turn those RNA intermediates back into double-stranded DNA, and then just like DNA transposons, it will insert itself randomly throughout the genome. And so these retrotransposons, as opposed to a DNA transposon, they're making copies, right? DNA transposons are cutting themselves out and moving around, but retrotransposons are making copies that move around. And so that's why there's so many of these elements throughout the genome, because they can duplicate. So while retrotransposons are able to easily make copies throughout the genome of themselves, it is possible for DNA transposons to do the same. And so as we can see here, during replication, we have a DNA transposon, that is in the DNA, and it gets replicated. As you can see here, there's 2 copies now at the DNA replication fork, and then it can cut itself out and move and insert itself to a piece of the chromosome that has yet to be replicated. So in that case, we now have 3 copies, right? Because this one's gone, and this one is intact, and then this one where it gets inserted will be replicated. So what we get is this end product down at the bottom here, where one strand has 1 copy, where it jumped in front of the replication fork, and the other strand has 2 copies, because it has the original copy that was left behind at the replication fork, and then that new copy that was inserted from right here. So it is possible for it to be replicated, but retrotransposons much easier to replicate because of the RNA polymerase mechanism of making RNA first and then reverse transcribing into cDNA and inserting. Let's look at the structure of LTR retrotransposons, which stands for long terminal repeats. And these are element-specific directed repeats, and they're about 250 to 600 base pairs long. And the central protein coding region, which I am marking right here, that encodes for reverse transcriptase, integrase,
and other retroviral proteins that are expressed that help to integrate these retrotransposons into the host's chromosome. So these LTR retroviral retrotransposons encode for all retrovirus proteins except for the envelope proteins that are required to actually make the virus. But the actual functional proteins of these RNA viruses are all encoded in these LTR retrotransposons. And so they behave like intracellular retroviruses. And so they're transcribed to form an RNA intermediate for generating retroviral genomes from integrated retroviral DNA. So this is very similar to the mechanism for generating retroviral genomes from integrated retroviral DNA. So all that being said, they're very similar to how RNA retroviruses work. So on the left side here, we have this LTR region, and this directs the cellular RNA polymerase to initiate transcription at the start site, which is right there. And then it extends to the LTR downstream, and it stops beyond the R site where it adds the poly A tail. Then this transcript is processed just like it is with any sort of mRNA within the cell, and it will encode for those retroviral type genes. Okay, let's look at how these mRNA are then converted back into DNA, which can then be inserted somewhere throughout the genome. Remember, these are jumping genes, quote unquote jumping genes, transposable elements that are able to be replicated and then moved throughout the genome. And so here we have our retroviral genomic RNA. And so this is the RNA that has been transcribed from that retroviral transposable element. And we get this tRNA that is specific for this retroviral RNA that binds to this PBS, which is the primer binding site. From there, we have transcription reading the RNA 3 to 5, and so we get a little piece of DNA that is transcribed that is attached to that tRNA. From there, this R and U5 site is degraded in the RNA, so it is missing. So we have this 3' overhang of DNA that has been attached to that tRNA. And now we have our first jump. So this complex of, let me circle it here, of tRNA and the synthesized DNA jumps over to the opposite side, and the R site binds to the 3' R site of the RNA. And from there, because the synthesis of DNA can only read the template in the 3 to 5 direction, this DNA is able to finish synthesis of our retroviral gene, as you can see here. From there, the rest, almost all, of the hybrid RNA that was our template is digested. And we have a almost completely single-stranded piece of DNA minus this little chunk of RNA that is still in the middle here. From then, the 3' end of the second DNA strand is synthesized. So now we are synthesizing this, reading our DNA strand to create double-stranded DNA in just this little section right here currently. From there, the rest of this mRNA is degraded, and we have double-stranded DNA on the 5' end of this blue template here. And then we have our second jump. So now this small piece of DNA, the second synthesized DNA strand, jumps back to where we started on the 3' end, and the PBS sites line up. And from there, we can synthesize in both directions to now have a complete double-stranded piece of DNA of our retroviral jumping gene transposable element. So let's talk about another way that genomic DNA is shuffled or recombined, and that is shuffling via recombination between homologous interspersed repeats. And in this case, we're going to look at the ALU repeats, which you've probably heard about in human genetic disease, but we're going to look at how these ALU repeats are used to shuffle DNA. So in this case, these ALU repeats are homologous, right? So they're the same sequence between the two. And so we can get homologous recombination based on the fact that these are the same sequences. However, this crossing over doesn't have to be one for one. And so this crossing over event here happens between two different genes, gene 1 and gene 2. So because of that, in between these two ALU repeats, we have two different exons. We have this blue exon and we have this green exon right here. And so what happens is gene 2 then will have that blue exon replace its green exon, so it's a longer coding sequence, and gene 1 will have that shorter green exon replace that longer blue exon that it originally had. And so what we end up having is these new novel combinations of pre-existing exons that can lead to genes with different new novel functions. We can also have transposons that help to reshuffle DNA and exons within DNA. And so first we have this DNA transposon that cuts itself out on these two flanking regions here, and within that flanking region we have an exon of gene 1. And so then when those transposons jump into another gene, they're also bringing an exon with it. And so instead of having these 4 small exons for gene 2, now we have those 4 small exons in addition to one larger exon that jumped into the gene. So this will change the conformation of the protein that is made from this gene and lead to novel function. And so this also can happen with this line system, which has an RNA intermediate. So what we have is line is a jumping mobile element that will also, because it has a weak poly-A tail here, will also incorporate this last exon. And so it becomes RNA, and then it transcribes back into DNA. Once it becomes that double-stranded DNA, it inserts, just like we saw with those retrotransposons. And so then within gene 2, all of a sudden, we have brought along this exon based on this. So let's talk a little bit about chromosome structure here. So here we have two electron microscope images that we're looking at. The first is chromatin that's isolated in some low ionic strength buffer, and it has this appearance of beads on a string. And as you can see, these little beads are actually the nucleosomes, and they're about 10 nanometers in diameter. And the string is the DNA in between those nucleosomes. So the nucleosomes are what wrap that DNA into a bundle or a circle. And then in the second image here, we have chromatin that is isolated in a buffer with a physiological ion strength. So it's not increased, it's what you would experience in the cells. And so here we have this condensed fiber that's about 30 nanometers in diameter. And these, at this point, there's nothing being replicated because it's all tightly bound into this chromatin structure here, and not, there's no way polymerase or anything can access that DNA. So let's look at the structure of the nucleosome. And so this is based on X-ray crystallography. So it's kind of this 3D model that an X-ray diffractometer is used to kind of determine what these structures are. And so the nucleosome is an octamer. So it's got 4 or 8 subunits, 4 on each side. And it's made of these histone proteins. And when they all come together, this octamer, we can have DNA wrap around it And generally here, DNA wraps around in about 147 base pairs of DNA are wound around this octamer. And when we need to access the DNA, that's when there is methylation or acetylation that happens to these histone tails that come off of these proteins and allow this DNA to kind of slide around that octamer and allow access for transcription factors and polymerases and things of that nature to either replicate or to transcribe that gene. Okay, here we're going to look at kind of a more meta view of chromatin and look at the actual chromatin fiber as opposed to the nucleosome. And so here we have a chain of nucleosomes. And I also want to point out here in our, when we looked at nucleosomes, we talked about the octamer, which is this circle right here. And that has 4 subunits and then 2 of each of those subunits, right? So you have 4 subunits on one side, 4 subunits on the other side that are identical to each other, and then they make up that octamer. But we also have this H1 histone, and the role of the H1 histone is to link that DNA between nucleosomes and kind of bind it to the nucleosome. So you can see here it's kind of like a little bracket that holds the non-bound DNA to the nucleosome. And so now looking at the chromatin fiber, there are 2 models for how these chromatin fibers exist. One is the solenoid model, and this is where nucleosomes are arranged in a left-handed helix. And so as you can see here, as it starts to be folded into the solenoid, you can see it's a single helix, right? And so that's the solenoid model. So the opposing model is the 2-start helix model, or the zigzag ribbon. And so in this model, it's more of a double helix. So if we were to trace this arrangement of the nucleosomes, and I'll use 2 different colors to accentuate it here, we can see that it looks more like DNA, and it's this double helix. So in the 2-start helix, though, I want to point out that it's not showing the DNA in between these histones, while the solenoid model is. So that's why they look a little bit different. But what you want to really pay attention to is the actual lines that I drew, the shape of these 2 models. So as I stated earlier, the way that histones are modified allows access to the DNA in those nucleosomes. And so this can occur through acetylation or methylation or phosphorylation or ubiquitination. And these tails have specific locations on them that are indicated on these diagrams here, depending on, so we have histone 2A, 2B, so these are 2 homologs of, or 2 monomers of that nucleosome complex, and you can see that the different sites labeled AC for acetylation, UB for ubiquitination, P for phosphorylation, and ME for methylation. All these different things can lead to whether or not access is permitted and the nucleosome slides, or if it's kind of locked in place. And so specific post-transcriptional modification combinations of these different chromatin regions influences the function by creating or removing these protein binding sites for like transcription factors and polymerase and things like that. And so these are important in their totality, their combinations of acetylation and methylation and ubiquitination, etc., as to whether these genes are actually able to be transcribed. So what's the point of wrapping up DNA into these histones and into this chromatin? Well, as said before, wrapping them up prevents access of transcription genes, and it also kind of protects the DNA when it's not being used. And so non-transcribed genes are less susceptible to DNases, which break down DNA, than active genes. And so in this experiment here, as an example, using chick 14-day embryos, there is a globin gene, or globin, excuse me, a globin gene that is expressed in 14-day embryos, but it's not expressed in undifferentiated lymphoblastic leukemia cells from chicks. And so, using this knowledge, they decided to do a test of access to genes. And so, nuclei from each cell type were isolated and exposed to increasing concentrations, as you can see here, of DNase. And so, nuclear DNA was then extracted and treated with this restriction enzyme, BAMH1, which cuts on either side of this globin gene. And then, after that, the DNA around the globin sequence should release a 4.6 kilobase globin fragment. So Southern blot analysis to look at the DNA from this extracted nuclear DNA was labeled and run on a gel. And so the transcriptionally active DNA from the erythroblasts was digested by DNase 1 in the nucleus, as indicated by the absence of the fragment. So because this gene was being transcribed in those 14-day erythroblasts, that DNase was able to access that globin gene and turn it into much smaller fragments. So it digested that globin gene at these fragments here, or at this concentration here of 0.05 micrograms per mil and above. Now, compare that to, and our control here is 0. So as you can see, when there is 0 DNases there, that globin gene's intact. But as you add DNase in increasing concentrations, it breaks down that exposed globin gene. Now, in the MSB cells, which are not expressing globin, or thus wrapped up in this condensed chromatin that we see pictured up at the top here, even when we treat it with the highest amount of this DNase, we end up with still our globin gene intact. And that's because it's wrapped up in that chromatin and protected and not exposed to any sort of protein, whether it's a protein that will transcribe it or a protein that will degrade it, like DNase. So looking at the cell, we have two different types of chromatin. We have heterochromatin and euchromatin. So heterochromatin is the inactive or condensed version, so it would generally be methylated, which restricts access to the genes. And so if we were to look at an electron micrograph, we would see that this heterochromatin is much darker because it's all condensed and all those proteins are piled in together. And then we have euchromatin, which has a lot of acetylation happening, which is allowing for the sliding of those nucleosomes and the uncondensing of that chromatin. And that appears a lot lighter because it's much more sparse, and that allows the nucleosomes to slide around, and transcription factors and polymerases and all the things needed to actually make RNA from that gene are able to get in and access that DNA at that point. So let's look at how methylation affects the formation of heterochromatin. So during the formation of nucleosomes, there are these tails, these histone tails. And these histone tails, like we talked about in the last couple slides, can be methylated or acetylated or ubiquitinated, etc. So in the instance of methylation, if there is a triple methylation at lysine number 9, and that is indicated here by this Me3, so that means there's 3 methyl groups at the 9th lysine of this histone chain. What happens is this HP1 protein will come in and it will bind to that methylated residue, the lysine, at position 9. And then what that does is these HP1 proteins all kind of bind together, right? So they contribute to this heterochromatin condensation by binding the histone H3 N-terminal tails that are trimethylated at lysine 9 and other HP1 molecules. So they kind of pull everything together, kind of act as glue to make this heterochromatin and really pull all that DNA and those histone proteins really tight together. And so there can also be spread of this heterochromatin formation. So if we look down here at B, we see that we have this HP1 that has bound to a methylated histone tail, that lysine number 9. And further down here, at the kind of end of this nucleosome chain, we have a methyl group that is not bound to an HP1 protein. And so what happens is this HP1 can recruit this complex of H3K9 and HMT, and it will methylate the next histone down. And once that occurs, that will allow HP1, which is not shown here, but I will draw poorly one, allow an HP1, this mega-sized HP1, to come in and bind to that methyl group. And then if you can imagine there being another histone here and another histone here, this can continue to occur where now that there is an HP1 right here where it previously wasn't, this H3K9 and HMT complex can then add a methyl group here, and which will allow another HP1 to come bind, and it will continue to go. And this can proceed continuously until we reach this boundary element. So this boundary element here is a position within the DNA that histones and nucleosomes don't form, but other proteins come and bind to prevent this continued chain reaction of binding up the nucleosomes with this HP1 protein. So if we take another step back and look even more meta, even more large scale, we have these formations of what are called chromatin loops. And so these red dots here are non-histone, non-chromatin proteins. And these proteins these could be, you know, 1 million or multiple million base pairs apart. But what they do is they pull this long strain of DNA into these tightly formed loops. And so when you look at a karyotype, where you see like the 23 chromosomes of a human, the 23 chromosome pairs, that they're really tightly bundled, and so we're getting closer to that. So there's a bunch of those little double helix shapes or single helix inside here, and the nucleosomes are even tinier little dots within that. And then add all together, these proteins pull the strands of DNA on each chromosome into loops. So here we can actually see the formation of these loops. We have a electron micrograph here, and as you can see, previously when we were talking about the beads on the string and things like that, we were at the nanometer scale, but now we're much bigger, we're at the micrometer. So we're zooming out, so to speak, on the DNA, and you can see the formation of these large loops. And so theoretically, somewhere in this area here, there'd be a couple proteins that are kind of pulling that loop together, and you can see multiple loops forming in this picture of the chromosome. Interestingly, during interphase, human chromosomes remain in these non-overlapping territories or domains in the nucleus. So they all have their own little neighborhood that they occupy, and they don't really overlap with one another. And so here we see part A is an interphase nucleus that's hybridized with fluorescently labeled probes specific for a sequence on each of those chromosomes. So the probe that labels 7 is a DNA sequence that is only found on chromosome 7. So as you can see here, obviously humans are diploid, so you get one chromosome from your mother, one from your father. So you have 2 copies of chromosome 7, and they are in those 2 locations. And you have 2 copies of chromosome 8, and they are this kind of purple color. And so there have been maps generated for all of the chromosomes, and as you can see in B here, they are non-overlapping segments where these chromosomes reside. So they're color coordinated here. So as you can see, chromosome 6 from your mom and chromosome 6 from your dad, they're in different locations here, but they're both yellow. So you can kind of see they're not necessarily next to each other, but they have their own territories, and they specifically do not overlap with other chromosomes. Okay, so looking at a metaphase chromosome here, you can see we have 2 sister chromatids. So a chromatid is one, let me switch to a better color here, is 1/2 of this pair, and because it's gone through replication. And we also have telomeres. So telomeres are the very end of each chromosome. So each chromosome has 2 telomeres. And then we have the centromere, which is kind of the center bundling point where spindle fibers attach. It's not necessarily exactly center because the P and the Q arms can be of differing lengths, but it's where the centromeres attach and pull the chromosome pairs apart. So let's look at the model for mitotic chromosome condensation or condensing. So here on part A, we have kind of this step-by-step progression of how DNA condenses into these chromosomes for replication purposes, or for cell replication. And so in phase 1 here, we have early prophase. So the DNA is all in strands, and it's able to be accessed for transcription purposes, gene expression. And then as we enter the middle of prophase for this mitotic event, you see that we start to get condensation or condensing of these chromosomes, and the blue indicates the kind of changes in large-scale chromatin folding, and the red is this SMC2 protein, which is a subunit of condensin, which is a protein that helps to bring everything together. And then by number 3 here, we have the late telophase. And as you can see, we're starting to see that structure of these chromosomes. And then stage 4 here is metaphase. So we have a TEM image here, so transmission electron micrograph of chromosomes, and it is stained with an antibody for this SMC2, which was the subunit for that condensin protein. And then lastly, we have this model. This is the hierarchical folding or axial glue model for the chromosomes as they're going through this process to become condensed chromosomes during metaphase there. And so as you can see, our normal 30 nanomolar chromatin fiber gets wrapped over and over and over again until it becomes larger and larger. It's almost like if you look at something like twine or yarn, where you have one little, these different fibers kind of wrapped up together to make it larger. It's not the greatest example because yarn or twine has multiple strings, and this is really just one giant string that's wrapped around it. But you get the idea. So it gets wrapped up into this big kind of glob that can be pulled apart during mitosis, or meiosis for that matter. So a karyotype is the chromosome complement of an organism. So you've seen this probably a lot, especially when we talk about things like Down syndrome or Klinefelters or Turners or things, chromosomal abnormalities. But how do we pair these up, right? So on the left, we have the full complement of chromosomes for a cell. And the way that we can identify each of these chromosomes is through fluorescent in situ hybridization, which is also known as FISH. And what these are is fluorescent kind of dyes that are specific to, or probes are specific to each chromosome. So we can change the color of those dyes, and since the part that attacks or identifies and binds with a chromosome are specific to each chromosome, we change the colors and the chromosome complement so we can identify them, right? So for example, purple here, the probes have a purple dye on it, and they're only specific for chromosome 2 in humans. And so they will label, all these FISH probes will identify chromosome 2. And from that, then we can go and see if there is an extra chromosome, and we can also identify whether it's a male or a female. So in this case, we have an X and a Y, so we know that it's a male. If we had 2 copies of the Y chromosome, which is this one right here, then the X chromosome. If we have 2 copies of the X chromosome, we know that it's a female. If we had 2 copies of the Y chromosome, we wouldn't know what the heck's going on, because that's not supposed to happen. But yeah, so this is how we identify males and females. Another thing we can do to identify males and females is when there is a female, they have these things in their nucleus called bar bodies. And so if we were to look at the nucleus, oops, this is kind of getting to be a bad image, they'd have this kind of bump that's sequestered to the edge of the cell, or of the nucleus. And this is an inactivated X chromosome. So you can look at the nucleus of males and females and visually see the difference because of the presence of this bar body. But we can also do it through the staining of chromosomes to identify X and Y chromosomes. In addition to identifying the chromosome count and male and female, we can also look for abnormalities in chromosomes due to translocation. So this is where a chromosome piece has been moved from one chromosome to another. So in this example here, we have myelogenous leukemia. That is a disease, and that is the result of what is called a Philadelphia chromosome. And I don't know if this was discovered in Philadelphia or if that was somebody's name, but it's called a Philadelphia chromosome. And this is where we have a shortened chromosome number 22. So this would be our normal chromosome 22 here, and an abnormally long chromosome number 9. So what is happening is, as you can see, number 9 takes a large chunk of 22, and in return, number 9 gives 22 this little piece down at the bottom. So we end up with a shortened 22 and a longer 9. And we call these DER9 and DER22. And so DER stands for derivative. So derivative chromosome 9 because it's partially 9, partially 22, and derivative chromosome 22 because it's partially 9 and partially 22 as well. But anyways, this leads to leukemia in patients that experience this. And there's also cri du chat, I believe it's pronounced, cry of the cat, which is a chromosome translocation, and other diseases that occur from breaking of chromosomes like this. So another thing that we can do with this FISH assay with chromosomes is to compare the evolutionary history of chromosomes. And so on the example on the left here between A and B, we see that there are 2 karyotypes. The top one is a tree shrew, and the bottom one is a human chromosome. So we have this, or human karyotype, excuse me. So we have this probe that is specific for chromosome 16 of the tree shrew. And if we use that on the human genome, we can see that there is this conserved piece of chromosome that throughout evolutionary history has stayed intact all the way up to humans. And we call that synteny, where we have this evolutionary preserved piece of chromosome. So the genes may have changed a bit here and there, but throughout evolution, that section in the tree shrew and that section in the humans evolved from a common ancestor. And if we were to look at this between primates and humans, right? So we know that primates and humans are very closely related evolutionarily. As you can see, we can make these maps that show the difference or the commonalities between the two. So here we have chromosome 1, which is the same between the two, and you can kind of see chromosome 3 in humans is what used to be chromosome 2 in primates, or is chromosome 2 in primates, so they've kind of switched around. And the reason that could be is because we arrange these in order of length, right? So chromosome 1 is always the longest, and the sex chromosome, or chromosome 23, is the shortest. So what used to be chromosome 2 in our common ancestor of primates, we could have lost a chunk of that as humans, and so it made it shorter, etc. A lot of rearranging and flipping of chromosomes and exchanging, kind of those translocations. A lot of that happens throughout evolutionary history. In addition to whole genome duplications and things of that nature. If you're interested in this sort of stuff, there's plenty of good courses on evolution. We're not going to get too deep into it. If you're at Tech, take Dr. Shepard's evolution class. We'll tell you all about this sort of stuff. But anyways, so we can use probes to kind of study this. And that's all I have for this chapter, so I'll see you in chapter 9.